A dog on this beautiful fall day, and as you have been out recently, as the seasons have started to change, you've probably noticed the beautiful color of many leaves that are on the trees. How does that happen? Leaves are normally green, right? Well, believe it or not, enzymes play a role in even the changing of the leaves, the seasons. So let's learn about enzymes. We'll find out how that works. Hey, let's take a look at enzymes together. Main idea: enzymes are catalysts for chemical reactions in living things. Now, how does that work? Well, enzymes are a catalyst. Generally, catalysts are substances that speed up chemical reactions. And how do they do that? Well, take a look. They decrease the activation energy, and that's how they increase the reaction rate. Let's take a look at this chart, shall we? In the solid line, you see that the reactants of this reaction are going to move forward, and a certain amount of energy is going to be needed in order for the reaction to move forward. And we can see this is the amount of energy that the products have in this reaction. Now, what enzymes will do is they will make it easier for these reactants to chemically react, and they're going to reduce that amount of energy that's needed for the reaction to go forward. And so therefore less energy is needed, the reaction can move forward at a much higher rate. Now, what does this all have to do with enzymes? Well, enzymes allow these chemical reactions to occur in our bodies and other living things under very tightly controlled conditions. They also are catalysts. The vast majority of those are proteins. And so there's literally thousands of chemical reactions that enzymes manage in our cells all the time. And because the enzymes have to work in tightly controlled conditions, homeostasis is critical for enzymes to move forward. If there's a disruption in the conditions within the cell, enzymes may not work at all. And the two key conditions that we're going to look at here are temperature and pH. As we mentioned a moment ago, because enzymes are proteins, and those proteins are held together in their particular three-dimensional shape by hydrogen bonds, changes in temperature and changes in pH can disrupt those hydrogen bonds that have the polypeptide chains folded in a particular way. As we mentioned before, proteins are made out of amino acids. Some of those are polar, and that's where the hydrogen bonds occur. Okay, so if you have hydrogen ions that are in a concentration that is foreign to what the enzymes are made to operate in, those hydrogen bonds can be disrupted, the whole protein can unfold and cease to function in the cell. Temperature as well can cause those hydrogen bonds to break and the protein unfold as well. So these two conditions are key in the function of an enzyme. Enzymes are specific to the compounds that they act upon as well. New term here, substrate. Okay, so what do we mean by a substrate? A substrate is a molecule upon which an enzyme acts. There's also a very specific location on the enzyme that's going to connect to the substrate. We refer to that as the active site of the enzyme. You can see the active site illustrated here by these notches in this illustration. There's an active site for this reactant that's in the gold, and the active site for this particular reactant is in the blue here. And you can see they're specific to the shape of those reactants. And so they fit together like a lock and key. So when the enzyme interacts with the substrate, it's going to weaken the bonds within the substrates themselves, and by binding to them and actually forcing a change, a three-dimensional change in the structure of that enzyme, and then causing it to weaken. And it's going to force the reactants to interact in a very specific way to cause them to bond to one another, and then the product is formed as you see here. Now, in this illustration, we have two reactants here to get one product. Of course, there are many different possibilities. You could have one reactant break down into two products. It really varies with the chemical reaction. While I have your attention, I should also point out that enzymes are going to, once they are there, they can be reused over and over again. They're not a reactant, so they're not going to be used up in a chemical reaction. They also need to be turned off at some point or not allowed to proceed. Once they're released into the system, they can't just operate independently. And so therefore, there are also molecules known as inhibitors that are going to either block the active sites that you see over here. Okay, so these are going to get blocked by a molecule that fits into that same shape, and that's known as a competitive inhibitor because it blocks the active site. There are also non-competitive inhibitors that will plug into an enzyme that will change the three-dimensional shape of the enzyme so that it doesn't proceed in controlling the reaction. It's deactivated. All right, great. So this is an overview of enzymes. All right, well, back to our question about the changing of the leaves. As you know, what makes leaves green in the first place is chlorophyll. That's a pigment that actually is part of the production of sugar in leaves. And keep in mind that leaves are really the kitchen staff for the tree. And during the busy time of the year for trees and making sugar, you need a kitchen staff to make all that energy that's going to get the trees through the year to meet all the energy demands that they have. And as the winter goes on, the tree lays that kitchen staff off. Well, part of that process and laying off the kitchen staff and getting the leaves to fall is dealing with chlorophyll. And there is an enzyme called chlorophyllase that actually that breaks down the pigment that's in the leaves. And then what remains, therefore, and this is going to vary from species of tree to species of tree, is you get the other colors that are present by other pigments that are in the leaves. Now, this maple leaf that I have in my hand has a lot of carotenoids, which are pigments that are similar to what you would find in a carrot. That's how they get their name. And so I'll leave it with you here in answering that question that we all come up with in the beauty of the fall. Very breezy day today. Well, listen, thanks for watching. Bye-bye.